Well, you don't have to tinker with the transcription factor as a protein itself. You tinker with these subtle aspects of space and time. Well, I turn it on in a few more cells,
Hmm.
or I leave it on longer, or those cells divide longer. That's going to change the size and shape of something in a viable way. That's the sort of stuff that variation is made of. So, what kinds of mutations have those properties? Those are generally mutations in the switches. Those are mutations in the non-coding parts of DNA that influence this choreography of how these genes work in space and time. And they're not catastrophic. And furthermore, these mutations, these switches, an individual gene, say one of these top regulator genes, it might itself have 10 or 20 switches associated with it. So, and a mutation in one switch has no effect on the other switches.
Hmm.
So if you think about a gene, for example, that might be involved in building bone, it might have a switch that turns on very late, say, for example, in the elaboration of the fingers. Okay. Well, tinker with that, and you might be changing the width and length of fingers.
Hmm.
Okay. But you're not changing the backbone, you're not changing leg length or things like this, right?